Hey guys, so after going through and looking at the Unit 1 exam, these are some of the topics that I found that in general class was still struggling with. And so in this video I'm going to go over those topics and hopefully clear some things up. So first thing I want to go over is the lac operon, and this was probably the section where people in general had the most difficult. There was quite a few questions on it, and it's probably one of the more difficult topics that was on this exam. So real quick, when we look at this, we have the two pictures here. We have the top one, well there's three pictures. This is just the lac operon without anything going on, so we're not really going to look at that one too much. But if we look at this first one here, where the repressor protein is bound to the promoter site and the RNA polymerase cannot bind, we would say this is off. So when we're referring to the lac operon and you hear on and off, when the repressor protein is bound here and the RNA polymerase cannot bind, that means transcription does not occur and we can't make those proteins. And remember, the lac operon, it makes these three proteins. They're enzymes. Remember, enzymes are just a type of protein, and those enzymes go and they help digest lactose, which is a carbohydrate in milk. So if the repressor protein is bound, the red thing, to the promoter site, the RNA polymerase, the green half circle, cannot attach there, and so these genes cannot be transcribed and the proteins are not made, and so we say it's off, right? The genes are not being produced, so it's off. On the bottom one here, as opposed to being off, this is on. We would say this is on. The lac operon genes are on now because, notice, the lactose here, the orange hexagon, has bound to the repressor protein. It's changed its shape, right? It's got a flat bottom here now, and it used to be like a little half circle so it could bind. Now it doesn't. It's flat, so it falls off. The repressor protein falls off. The RNA polymerase binds to the purple promoter site. And now you can see this arrow here, transcription begins, the RNA polymerase moves down here, it transcribes these three genes, and then they'll go into the cytoplasm for translation, and we'll make those proteins, we'll make those enzymes, and those enzymes will then digest lactose. Now another important thing to know is that the lac operon is only on when lactose is present. Right, so it's only on if lactose is present, and that makes sense because if it was on all the time, it would be producing these proteins all the time, and that would be a waste of resources for the body. If there's no lactose present in the body, if there's no lactose here that's in the body, then it's a waste to make these three proteins because they're just going to sit around and do nothing, and our body wants to be as efficient as possible. And so remember, for transcription to take place on the lac operon, this repressor protein must fall off. It has to fall off, right? If it doesn't fall off, then the green half circle RNA polymerase cannot bind and there'll be no transcription. So that repressor protein has to fall off. Has to fall off. Okay, now another misconception that I saw in the exam was related to biotechnology and specifically DNA recombination. And so remember in class we talked about the insulin gene, and actually I'm going to throw up a diagram here real quick. Ah, magic, the diagram has appeared on the screen. Okay, so here's our diagram that we looked at for DNA recombination, right? And remember, DNA recombination, specifically we used insulin as the example, is you take a gene from one organism. In our case, we'd take the insulin producing gene from a human, right? Maybe this is the insulin gene. And then we take a plasmid from a bacteria or some other organism. And plasmid, remember, is just a small circular piece of DNA. So this would be a plasmid in a bacteria. We cut it open because we have to put this gene in this plasmid. And you can see we do it right here, right? Now that gene, the insulin gene, has been inserted into the plasmid. And then we just stick that back into a bacterial cell, and then we let it reproduce. And then we get a whole bunch, so after this cell, we'd let this reproduce, and then we get a whole bunch. And this is like one of these, a whole bunch of bacteria cells. Right, this bacteria cell is producing. There's a whole bunch of these now, and they all have this insulin gene inside of those cells now because we put it in there, and then this one cell, this is the original cell, it's reproducing into all these other cells, and they're all producing insulin. Right, so now all these cells are producing insulin, and then we can take that insulin and we can use it for ourselves, for people with diabetes. So the point of DNA recombination, of inserting that DNA from one species into another, is to make the second species, to make the second organism produce proteins. Insulin's a protein, so that's what we want. We want these to produce proteins. We are not creating a new species. These bacteria here with the insulin gene are not a new species. We do that because we want them to produce specific proteins, and in our example, we want them to produce insulin. Insulin's a protein, and that's what we want them to do, so that's why we put the genes from one organism into another. And then one last thing I want to go over deals with development and the expression of genes at different times during development. There was a question asked that was saying, as humans are developing from an infant to a child to adult, then there's a different kind of hemoglobin in their blood, and hemoglobin is part of your red blood cells. It's the part that carries oxygen. And so remember, we need to know, and I think all of us know this, that all cells in your body contain all of your genes, right? Muscle cells and bone cells contain the same genes, just some of them are expressed and some of them are not. Right, so all cells contain all genes. All your cells have the exact same DNA. And that doesn't go just for that, not only goes for types of cells, for muscle and bone cells, but also at points throughout your life. So your cells, when you're a baby, when you're an infant, have the exact same DNA as you do when you're 80 years old. That doesn't change throughout your life. So you don't gain or lose genes, right? Your blood cells, when you're a baby, have the exact same genes as you do when you're an adult. Just some genes are turned on when you're an infant, and some different genes are turned on when you're an adult. Right? So it doesn't matter how old you are, you always have the same genes, just they're expressed at different times. So some genes will be expressed when you're an infant, some expressed when you're a child, and some expressed when you're an adult. But yeah, you don't gain or lose genes, they'll just be expressed at different points in development. And so those are the three main topics that I wanted to go over here. And if you'd like to retake the test, then you may do so after watching this video.